projection. Is it? Size of the slide, maybe. So the endocrine pancreas. Where is the pancreas situated?
The biggest station.
In the abdomen. Where?
Below the stomach.
Below the stomach, and very much adjacent to the duodenum.
Yeah.
Isn't it? Duodenum. Why? Because the contents of the pancreas, they are what? Lights? Where is the switch? Outside? So, the pancreas, they are basically divisible into two. One is the exocrine part of pancreas, another is the endocrine part of pancreas. Exocrine pancreas is, the terms are self-explanatory. Exocrine and endocrine. Endocrine is concerned with synthesis and secretion of hormones, whereas exocrine is concerned with
Enzymes.
digestive enzymes and digestive juices. So, as they are concerned with digestion, they have to secrete their juices directly into the GIT. And where is the GIT? Not in the stomach. So, they are secreting, because in the stomach, the functions of stomach, one of the important functions of stomach is to simply store the food and churn it up. Make it smaller particles, ultimately making time out of it. So objectives, etc., you already know what we have to look for in the pancreas. So this is the pancreas, and this is the duodenum, and just above here is the stomach, like that's so rightly said. Now the pancreas is physically divisible into three: the head part of the pancreas, the body, and the tail part of the pancreas. And as you can see, the pancreatic duct, which is coming over here, and here is the point of its opening inside the duodenum. Exocrine part, digestive functions; endocrine part, synthesis and secretion of hormones. Basically two hormones, insulin and glucagon. So when we are talking about insulin and glucagon, basically what we are doing is, we are talking about blood glucose level and its regulation in the body. That is the primary thing, because these hormones, insulin and glucagon, they are having opposing functions to one another. We will study in depth about functions of insulin, and we will cover glucagon very briefly, because it is just opposite to insulin. So if you understand everything about insulin and what it is doing, it is very easy to just apply the reverse to glucagon. Pancreas also secretes some other very small quantities of other substances, like amylin, somatostatin, and pancreatic polypeptide. When we are talking about synthesis and secretion, we have to look up at least some histological part which is related to physiology, which are the cells, etc. So, in the pancreas, there are two major types of tissues. The exocrine pancreas is containing the acini, which are responsible for synthesis and secretion of the digestive juices, the digestive enzymes into the duodenum. And the second type, which you can see over here, are the islets of Langerhans. So these structures that you see over here, these are the acini, pancreatic acini. And what do you mean by islet? Small island. Isn't it? Hence, as you can see a group of cells in between the acini, which appear like small, very small pieces of islands over here. That is why they are called as islets of Langerhans. Now these islets of Langerhans are about 1 to 2 million in the pancreas, and they are organized around small capillaries into which their cells secrete the hormones. These are the capillaries. And because, as you know, hormones are directly secreted into the bloodstream, so those are the capillaries in which the hormones are being secreted. The islets, they contain three major types of cells: alpha, beta, and delta. Out of this, if we look up, the 60% out of these cells is the beta cells. 25% are the alpha cells, and the remaining 10% are the delta cells. 60% beta. What are they responsible for? Naturally, they are responsible for insulin. Insulin and amylin. Alpha cells, glucagon. Delta cells, somatostatin. There is one other type of cell, the PP cell, and which is responsible for secretion of the pancreatic polypeptide.
The percentage of cells different from the listener.
Different, yes.
So, in the example,
Histological percentage. Okay? Because they are supposed to be more correct. We are physiology people. We are concerned with the functions, not with numbers. Anyway, even if you see physiology books, different physiology books, you will find different numbers. So it's always there. From author to author, from researcher to researcher, numbers are different. You have to remember the main point that delta cells are maximum. Second comes the beta cells are maximum. Second comes the alpha cells, and least in number are the delta cells. That is how you have to remember mostly. Now, actions of insulin. Whichever hormone is acting, it has to act via receptors, isn't it? It has to bind with its receptor first. Where are the receptors present? Do you know about hormone action? Yes. The receptors can be present on
cytoplasm or cell membrane or nucleus.
On the cell membrane, very right. Or in the
Cytoplasm or nucleus.
Or in the nucleus. Isn't it? These are the three areas where the cell receptors can be present. Here, the insulin receptors are present in the surface of the cell membrane. Okay, and the type of insulin receptor, as you can see, this is the cell membrane. The type of insulin receptor is such that half of it or part of it is embedded. Can I say embedded or penetrating through the cell membrane, whereas half of it is outside the cell, outside the cell membrane. So this portion is outside. So it's a tetramer. Tetra means 4. The insulin receptor is a tetramer having 2 alpha and 2 beta units. The alpha units are completely outside the cell, whereas the beta subunits, they are the portions which are penetrating the cell membrane. Easy to understand? Okay. Now, insulin, this blue colored structure is insulin. When insulin comes and binds with, obviously it will bind with which portion? Alpha units, not the beta, because the beta are penetrating inside the cell membrane. So it binds with the alpha. As soon as there is binding of insulin with the alpha, there are changes in the beta. This change is phosphorylation. And that is called as autophosphorylation, as it is written here, autophosphorylation. Now when there is autophosphorylation of the receptor, you must have studied that whenever a ligand can be a hormone or any other substance, isn't it, which is binding with the receptor. So commonly it is called as a ligand, but here we are talking about a specific hormone which is insulin. So let's call it as a hormone. So when a hormone, insulin, is binding with its receptor, it is supposed to change the inside of the cell. Whatever is going on inside the cell, isn't it? That is the effect it has to have. So similarly here also, and to bring about this particular change, what will it do? It will either activate some enzymes or inactivate other enzymes. Isn't it? That is the effect that the binding of the hormone with its receptor is supposed to carry out. Similarly here also, there are a number of enzymes within the cell which are activated, whereas some enzymes which are not needed at that particular time to be activated, they will be deactivated. So depends upon the need at that particular time. Say for example, we have eaten a meal which is rich in carbohydrate. So naturally, the blood glucose level will be much higher as compared to the blood protein level or the blood fat level, isn't it? Or conversely speaking, if we eat a meal which is rich in meat, for example, okay, so the protein level will be higher and the carbohydrate level will be very, very low. So depending upon what is higher, those particular enzymes have to be activated inside the cell. And this phosphorylation of the beta subunits, which is also called as autophosphorylation, causes these changes, as you can read over here. Autophosphorylation of the beta subunits of the receptor activates the local tyrosine kinase inside, tyrosine kinase, which in turn causes phosphorylation of multiple other intracellular enzymes. Okay? The net effect, net means the final end result, is to activate some of these enzymes while inactivating others. Why? Depending on the need. In this way, insulin directs the intracellular metabolic machinery to produce the desired effects on carbohydrate, fat and protein metabolism. Now here you see fat synthesis, protein synthesis, glucose synthesis. Is this what is going on inside the cell? Yes. Yes. Why synthesis? Why we are calling it a synthesis? As you know, proteins, or let us say amino acids are the body's building blocks. They come together to make proteins, and the proteins are basically building up our body. How this is happening, we will see in detail later on. But one of the important functions of insulin is to carry this out. Okay? You will see that. Are you clear about actions of insulin? Okay? Let us go ahead. Now, functions of insulin, physiological effects or functions of insulin. We can talk about general effects, okay, and then we will be more specific, like on the cells direct. General means what? On the global human metabolism. Global human metabolism is on the metabolism of the whole body. Global yani metabolism of the whole body, generally speaking. So generally speaking, what is the action of insulin? Number one, control of cellular intake of certain substances. Most prominently glucose in the muscle and adipose tissue. Why most prominently? Because about 2/3 of our body is muscle and adipose tissue. 2/3. Second is increase of DNA replication and protein synthesis via control of amino acid uptake. And third is modification of activity of numerous enzymes, like we have seen just previously, action on a particular cell. So modification of activity of certain enzymes. So this is an effect which is general. Now let us go down and be more specific. Actions of insulin, it can be direct or indirect on the cells of the human body. 1, 2, 3, I have tried to make it up to 12 only. These are also other functions. These are interesting functions of insulin which are there for you. And it is very easy to remember all of them. Okay, I have just made it out into a longer list, 10 to 12 functions like that. But it is very easy to remember because basically it is having effect on metabolism. Okay, inside, like we have seen in the previous slide, all the 3 types of metabolism. So either it is increasing that or decreasing that. So as you will see, increased glycogen synthesis. Now what is glycogen? It is a form in which glucose is stored in the liver or muscle. So increase in the rate of glycogen synthesis, conversion of glucose into glycogen. Insulin forces storage of glucose in liver and muscle cells in the form of glycogen. Number 2, increased potassium uptake. Now, what is this glycogen? Glycogen is a substance which is basically increasing the osmotic pressure inside the cell. Okay, that particular change inside the cell causes more and more potassium to move to maintain balance. Okay, increased potassium uptake. You are late, both of you. Please take permission from Dr. Main. Forces cells synthesizing glycogen to absorb potassium from the extracellular fluids. Okay? What? What forces the cells? Like I have told you, change, okay, inside the cells, particularly the liver cells and the muscle cells most of the time. And as you know, the muscle cells and the adipose tissue cells, like we have seen, they are comprising about 2/3 of the body. So electrolyte level, to some extent, is also controlled in this indirect way. This is not a direct function of insulin. So that is why it is written here indirect and direct. So this is an indirect function of insulin. What is that? What is one of the indirect functions? It causes increased potassium uptake by the cells. Indirect. Direct effect, decreased gluconeogenesis. What is gluconeogenesis? Yes, from different substrates, sometimes when the body is deprived of glucose, not enough glucose is present in the body, then the body will try to produce glucose itself. And this it does using different substrates. Okay? Here again the function of insulin is there. Naturally, what does insulin do? What is the main function of insulin?
Decrease glucose.
To move glucose from the blood into the cells. Isn't it? That is the main function. So it is very easy to understand that it will inhibit or it will decrease gluconeogenesis. Why? Because its function is to remove glucose. And gluconeogenesis is adding glucose into the blood.
Yes.
Correct? So it is very easy to remember like this. The main function being to remove glucose from the blood. Naturally, what will be the other function? To inhibit or decrease all those things which will cause glucose level to rise in the blood. Isn't it? So one of them will be to inhibit or decrease gluconeogenesis. What will be the other? Can you tell me now? I did not go into it.
It's glycolysis.
Glycogenolysis. Isn't it? It will prevent or inhibit glycogenolysis. So it's simple if we just go on like this. Okay, increased lipid synthesis, as we have seen. Isn't it? Increased lipid synthesis. How it does that? Insulin forces the fat cells to take in blood glucose, which is converted into triglycerides. How? We will see in the future slides. Increased esterification of fatty acids forces the adipose tissue to make the neutral fats, that is triglycerides, from the fatty acids. Please take permission from Dr. Main because you are late. It is 8:20 now. Okay? We should. I think there is a key here. Okay, I just locked it. I did not know that the key is there, so that we will not be disturbed again and again. Okay. Decreased lipolysis. Naturally, again, just like we have seen its action on insulin, isn't it? Insulin's action on glucose. So if it is doing one thing, the other which is promoting will be inhibited. Similarly here, there is increased lipid synthesis and esterification of fatty acids. So naturally there will be decreased lipolysis. Like there was decreased glycogenolysis. Correct? So decreased lipolysis forces reduction in conversion of fat cell lipid stores into blood fatty acids and glycerol. Next is decreased proteolysis. Again, why? Because it is basically involved in protein synthesis, yes, isn't it? So it will cause decrease in proteolysis, decrease in the breakdown of protein. Eighth is decreased autophagy. Who is that? What do you mean by autophagy? Yes. Phagy is the word coming from, as you know, we use it in different places like phagocytes, phagosomes, phagocytosis, isn't it? What is that, phagocytosis? Phagocytosis is the process of engulfing or eating of bacteria, for example. Here, autophagy, the cell is digesting itself or eating itself. But It's not like that. I mean, it doesn't digest or eat itself completely. The thing is, during metabolism, during the normal processes which are going on, some of the cell organelles may be damaged. And because of the damage to the cell organelle, the cell itself, now who will come from outside and know that this cell organelle is damaged? The cell itself has to do the correction. So what it does, it causes lysis and destruction of the damaged cell organelles. This process is called as autophagy.